that, which is quite interesting. So there you see a developing neuron actually migrating along a glial cell guy wire, performing this radial migration. And if you look kind of closely at this picture, no, don't do that to me now. Thanks a lot. Great. Anyway, I was going to say, if you look kind of closely at that picture, you'll see that there are sort of some fibrous-looking projections. I hope I haven't lost this recording. Okay. I may have to... Okay, I'll have to restart it. Okay. Yeah. Last Thursday, I forgot my computer because I was trying to remember a million other things that day, and I used a departmental computer that didn't have Tegrity on it. So I'm sorry, there's not one. The slides are what we've got for that one. I can, if you find that you feel like you're missing too much, send me an email and I'll post like I did for that section that was missing the sound. I posted some lecture notes. Did anyone use those? No? Okay. Okay. I said in class that I would do it, and I did it, but I didn't know if anybody had even, found them or used them. So if you find, you need them, I'm happy to do that. It's a little bit of time for me to go through and, write them out, but I'm happy to do that. If you feel like you need it, it's no problem. Send me an email. Okay, sorry, I've got to go back and restart because I've got a couple of things I wanted to. Oh, there we go. Okay. Okay, so anyway, you can sort of see there are some fibrous projections coming in the front of this cell, which see them kind of flashing a little bit. It's not a great picture of them, but those are the cell. If you look back to this picture, those are these. Basically, these little projections that migrating cell is sending out, grabbing onto the glial cell guy wire and pulling itself along the guy wire to translocate. And they're represented by these kind of squiggly lines that you see here.
Shortly after fertilization, the fertilized egg or zygote begins dividing. By about 4 days after fertilization, a solid ball of cells called a morula forms. The cells continue to multiply, and by day 5, the cells separate into two groups, the inner cell mass, which will become the embryo, and an encompassing sac called the trophoblast, which will develop into the placenta. Trophoblast cells secrete fluid, creating a cavity, the blastocele, with the inner cell mass at one end. At this stage, the mammalian embryo is called a blastocyst. The cells of the inner cell mass eventually form two layers, the epiblast and the hypoblast. The epiblast is thought to contain the cells that will generate the actual embryo. Critical to the early development of all vertebrate embryos is the process of gastrulation, which occurs between days 13 and 19. Gastrulation results from the movement of cells toward the midline and then forward along the midline, forming a groove called the primitive streak. The end result is the production of the three primitive cell layers, the endoderm, the mesoderm, and the ectoderm. The nervous system develops from the outer layer, called the ectoderm. As this cell layer thickens, it becomes a distinct epithelium called the neural plate. Uneven rates of cell division form a groove called the neural groove that will become the midline of the embryo. Ridges of ectoderm continue to bulge on both sides of the midline. The tops of the neural ridges eventually come together, forming the neural tube. The interior of the neural tube becomes the fluid-filled cerebral ventricles of the brain, the central canal of the spinal cord, and the passages that connect them. By about 24 days, the three major subdivisions of the brain are discernible. These include the prosencephalon, or forebrain, including the telencephalon and diencephalon, the mesencephalon, or midbrain, and the rhombencephalon, or hindbrain. The progenitor cells of the neural tube are known as neural precursor cells, or neural stem cells. This represents the first stage of neural development, neurogenesis, when undifferentiated cells undergo mitotic divisions to produce either new stem cells or neuroblasts that will eventually differentiate into neurons. The cells undergo a stereotyped pattern of cell movements as they progress through the mitotic cycle. Eventually, the dividing precursor cells form a closely packed layer of cells called the ventricular zone. Eventually, some cells leave the ventricular zone, forming a second layer of cells called the marginal zone. Later, an intermediate layer develops as the wall thickens. Cells in the intermediate zone will then begin to differentiate into both neurons and glia. As the number of cells increases during the period of neurogenesis, the newly formed cells must move even greater distances. The massive movement of nerve cells to establish distinctive cell populations represents the second stage of neural development, cell migration. Cells do not move in an aimless, haphazard manner. Cells in the developing cortex move along the surface of a particular type of glial cell called radial glia. Like spokes of a wheel, these radial glia cells extend from the inner to the outer surfaces of the emerging nervous system. The radial glial cells act as guide wires, and the newly formed cells creep along their processes.
You can kind of
Once cells reach their appropriate destination,
see the
first
successive
stage
layers
of neural
coming.
development begins, differentiation. At this point, the cells begin to use or express particular genes. This allows the cell to acquire the distinctive appearance and functions of neurons characteristic of that particular region. As the process of differentiation proceeds, the fourth stage of neural development begins, process outgrowth. The biggest change in brain cells early in life is the extensive growth of axons and dendrites and the proliferation of synapses. The neural environment also greatly influences nerve cell differentiation. In vertebrates, young neural cells seem to have the capacity to become many varieties of neurons, and the particular type of neuron that a cell becomes depends on where it happens to be and what its neighboring cells are.
Okay, so you can get anything from a vastly, diversely branched Purkinje cell in the cerebellum to a very simple retinal bipolar cell, which really only has two projections. And all of that level of differentiation is dependent upon the environment that a cell finds itself in when it reaches its final destination. So once it makes that decision, it doesn't go back. Okay, let me just, I think I have one I want to show you there. Okay, I'm going to wait a sec to say a couple more things here. On the growth cone,
One of the most extraordinary features of nervous system development is the ability of growing axons to navigate over long distances through complex embryonic terrain to find appropriate synaptic partners. The growth cone, a specialized structure at the tip of the extending axon, or dendrite, is a highly mobile structure that guides the extension of the growing tips of neurons. The primary morphological feature of the growth cone is a sheet-like expansion of the growing axon at its tip, called the lamellipodium. When growth cones are examined in vitro, lamellipodia can be seen clearly, as can numerous fine processes called filopodia that extend from lamellipodia. Filopodia rapidly form and disappear from the terminal expansion, like fingers reaching out to sense the environment. The lamellipodium and filopodia are distinguished from the axon shaft by different cytoskeletal molecules, and growth cone motility reflects rapid, controlled rearrangement of these elements. These elements include actin, which regulates changes in lamellipodial and filopodial shape, and microtubules, which are responsible for the elongation of the axon itself. Actin is the primary molecular constituent of a network of cellular filaments that are found in lamellipodia and filopodia. Actin is found in two basic forms in the growth cone and axon: freely soluble monomers in the cytoplasm, also called globular or G-actin, and polymers that form filaments, also called F-actin. Various actin-binding proteins regulate the assembly or disassembly of actin filaments. Tubulin is the primary molecular constituent of the microtubules that run parallel to the axis of the axon. Like actin, tubulin is found in two basic forms: freely soluble monomers in the cytoplasm and polymers that form microtubules. The dynamic polymerization and depolymerization of actin at the membrane of the lamellipodium, as well as within the filopodium, sets the direction of growth cone movement, in part by generating local forces that orient the growth cone toward or away from local substrates. Actin subunits are added at one end and are removed from the other. Similarly, the polymerization and depolymerization of tubulin into microtubules consolidates the direction of movement of the growth cone by stabilizing the axon shaft. The interface between the actin and microtubule cytoskeleton is particularly important for regulating the balance of active growth versus stability in a growing axon.
So there you can see the molecular mechanisms by which this growth is accomplished in